Welcome back, AP Bio, as we talk about 11.3 and looking at some inheritance patterns in genetics. So we've been talking about dominance with our Punnett squares. So for example, a dominant allele in a genotype will lead to that dominant phenotype being expressed. So these dominant alleles will be written out with the capitalized letter or the big version of the letter. And we know that anytime there's a dominant allele present, that trait gets expressed for that character. So if you are homozygous dominant, big A, or heterozygous, big A, little a, the dominant allele will win out, and you end up expressing that dominant trait. For the recessive trait to be expressed or seen, you need to be homozygous recessive, or both alleles on the homologous chromosomes need to be that recessive trait. So this is what we've been talking about so far. Like, for example, when we talked about the peas in Mendel's experiment, yellow was dominant to green. So when he crossed the yellow and green true breeding line homozygous peas together, all the offspring were yellow because they were heterozygous, so they showed the dominant trait. Now, this isn't always going to be the case. So when we explore some of these other possibilities, I want to explain why this happens at a molecular level. So remember that alleles are alternate forms of a gene, and genes are the code in the DNA to make a protein. So if that code gets changed slightly, we're going to build a slightly different protein, or potentially no protein at all. So say there's an enzyme that converts pigment in flower petals to the color purple. So if there's a mutation that causes that protein to be made poorly, it doesn't quite come together, it's non-functional, or perhaps there's a mutation that causes it to not be made at all, that enzyme isn't going to exist, or it's not going to function properly, and it's not going to convert that pigment to purple. So that's why we can see that there's a dominant and recessive allele. Now, sometimes when you have that dominant trait, when it's heterozygous, half of those alleles are making a working protein, half are not, and so that working protein can still convert it to purple. It still does the job, and that's why you see expression of the dominant trait, because that functioning protein is still making the trait happen. But sometimes that dominant protein in a heterozygous trait just isn't enough. So, for example, in incomplete dominance, this is a heterozygous form where that heterozygous genotype produces kind of an intermediate phenotype that's halfway between the recessive and dominant phenotypes. So heterozygous is almost dominant, but not quite. So imagine that same thing where pigment is being changed over from white to red by an enzyme. If you have half the amount of enzyme, maybe some of the pigment gets converted, but not enough. And so because it's only going to convert half the amount of pigment, those petals are going to appear like pink instead of a bright red, because there just isn't enough pigment made to really bring that bright red color. Incomplete dominance is usually when there's a non-functional protein for recessive. Another great example is your taste buds. If you have a dominant trait, those taste buds taste really strongly. If it's recessive, you can't taste a certain flavor. And if it's heterozygous, you can sort of taste it, but it's not overwhelming because half of your receptors are working, half are not. So you only get a weak signal being sent to your brain. Another example of this is codominance. So this is where both traits are producing functional proteins, but they're going to result in different traits. So for example, an enzyme that produces white pigment and an enzyme that produces red pigment. And so they're both competing for space. And so you'll see both of them. It's not a blend of red and white to make a pink, but instead you see some white and some red based on where that pigment is being produced. So both traits are being expressed at the same time because both proteins are functioning. It's just that mutation in the code causes the protein to do something slightly different. So that's codominance. Another thing that we're going to talk about is multiple alleles. So most things in your body are not as simple as two alleles, dominant, recessive, and those are your two options. Think about hair color. There's a lot of different variations of hair color. Even just thinking about just brown hair color alone, there's different shades and gradients of it. And so typically in mammals, fur or hair are controlled by multiple alleles that are working in concert to create all sorts of different patterns, like you can see here with the rabbits. So when you have more than one allele, what you'll do is use a capital and lowercase letter to denote the dominant and recessive character. And then you'll have superscript or exponent alleles that show the different allele possibilities. So this will determine if it's dominant or recessive. So here you can see there's a dominant trait for brown fur. And then there's several different variations of the recessive trait. Your blood is the same exact way. So A blood type produces a A antigen that's read by your immune system. B alleles produce a B antigen. So if you have both the A and B alleles, you end up with both being expressed. So here we have codominance and multiple alleles. And then the recessive form O produces no antigens. So you can tell that because this is recessive, if you have like a mixed genotype of A and O, because O produces nothing and A produces only A, you'll still have an A blood type. Same with B. If you are producing B with half your alleles and nothing with your other half, still going to show B. And then you have this codominant where you're producing both A and B. So here, because the protein is immunoglobulin, we're going to use the capital and lowercase letter I. And then in our superscript or exponent, we're going to use the letters A and B and O to denote our different alleles. So what does this look like in a Punnett square? Well, you're still going to do the same concept. So IA drops down, IB comes over, and you have IA, IB to show that we have like a codominant AB blood, some A blood, some B blood, and some O blood possibilities. So just getting used to seeing what that exponent notation means. This is still just one allele, even though there's two different letters there. Last thing I want to talk about today is epistasis. Epistasis is when you have a pathway of multiple genes working together to produce a trait. And so you can think of it like a series of dominoes. The first domino gets tipped over, that's going to hit the second domino and the third domino. We've talked about with protein signaling when you have multiple steps in a pathway. And so this is the same kind of concept. If you remove a gene in the earlier steps, then the later steps can't happen, because if this is an enzyme, enzyme 2 here, it's going to work on this, its substrate is going to be the product of enzyme 1. So if enzyme 1 doesn't exist, enzyme 2 never gets its substrate and it can't perform its function. So a great example of this is mouse fur color. There's a gene that is going to activate pigment, and then another gene that's going to convert the black pigment to a brown pigment. And so we have black as recessive and brown as dominant, or agouti as it's known for mice coloration. But all of this only takes place if you have a dominant C allele for albinism. So if it's recessive, this gene isn't working to make the pigment at all, we end up with a albino mice no matter what gene 1 says. Now, if we get beyond that, we can get to the different possibilities here. So sometimes we're going to have, no matter where one gene masks another one. And I just want you thinking about it in a pathway, stepwise sort of organization. All right, that's all we have for 11.3. See you next time.